Usually, a medical diagnosis is pretty binary. You know, you break your arm, the x-ray shows a jagged white line, and the doctor just points at it, broken or not broken.
Right, it's very definitive.
Exactly. But as you know, being a clinical embryologist preparing for ESHRE, the ART laboratory, is basically the absolute definition of diagnostic murky waters.
Well, absolutely.
I mean, you're looking down a microscope at a living cell, trying to predict a human life based on, like, shadows and textures and timing. It's wild. So today, we are doing a deep dive to uncover the unbroken chain between that microscopic biology and the patient's ultimate clinical outcome.
Yeah, we are taking everything from the intracellular machinery straight to the lab bench.
Right.
And that's exactly the mission here. If you've been following this, you know we've spent a vast amount of time in the microscopic weeds. We've mapped out the architecture of the cell, the fluidity of the membrane.
The power output of the organelles.
Yeah. Yes, all of it. The tensegrity of the cytoskeleton. But for the ESHRE exam, and frankly, just for running a successful ART laboratory, you have to move far beyond just memorizing those isolated facts.
A flashcard definition of a mitochondria doesn't really help you when a patient has, zero fertilization on day one.
Exactly. It's useless in that moment.
And I think that's where a lot of embryologists hit a wall. It feels like we've been in a garage building this high-performance engine piece by piece. We spent hours studying lipid bilayers and polishing pistons. But this part of the journey, this integration phase, is about putting that engine on the racetrack.
I like that analogy. It's a high-stakes transition. The ESHRE certification demands practical application. You have to take pure molecular biology and actually use it to mitigate laboratory stressors or apply morphological grading.
Right, probabilistically ranking embryos.
Exactly. And even conduct patient counseling. Every single clinical decision you make at the bench draws entirely on the intracellular biology we've been covering.
So, to get us grounded here, what does this integration phase actually look like cognitively? Because we aren't just memorizing textbooks anymore.
No, not at all. It requires adopting a very specific mindset, which ESHRE structures into 4 ascending levels of cognitive demand. Level 1 is simply describe. You just need to describe the main intracellular structures. What is the plasma membrane? What is the cytoskeleton?
Okay, so just the baseline vocabulary.
Yes, but then level 2 is relate. You have to relate those structural features to their baseline functions. So you don't just know what a phospholipid bilayer is, you relate its amphipathic nature to selective permeability.
Makes sense.
You don't just identify the cristae of a mitochondria, you relate that massive surface area to the electron transport chain and ATP production.
Okay, so structure relates to function, but that still feels a bit like, undergraduate biology. Where does the clinical embryology part start?
That starts at level 3, link. You must link those general organelle functions to the highly specialized biology of human gametes and embryos.
Give me an example of that.
Well, for instance, linking mitochondrial mass specifically to the developmental competence of an aging oocyte. Or linking the smooth endoplasmic reticulum not just to general calcium storage, but specifically to the highly orchestrated calcium oscillations required at fertilization.
Oh, wow. Okay. And the final tier?
Level 4 is integrate. This is the summit. You have to integrate structure and function to explain how specific microscopic alterations affect the entire developmental timeline.
So following the dominoes all the way down.
Exactly. If a structural defect is present, how does it alter cleavage dynamics? How does it impact compaction on day 4 or blastocyst formation on day 5? And ultimately, how does it change the ART outcome?
I have to push back a little here, just practically speaking. It's one thing to understand that conceptually, but how does an exam actually test integration in a high-pressure multiple-choice scenario? How do you test if I can integrate the function of an actin filament?
They test it by giving you complex failure scenarios, not vocabulary matching. To survive those questions, and honestly, to survive in the lab, there's one core integrating question you must constantly ask yourself. This needs to be your mental anchor.
Okay, lay it on me.
If this structure is damaged or absent, what fails, and what does the embryologist observe?
If the structure is damaged or absent, what fails, and what does the embryologist observe?
Okay, let's unpack that. I want to test that logic right now. Give me an example of reasoning backward from a clinical observation.
Sure. Let's take severe blastomere asymmetry on day 2. The observation is that one cell is massively larger than the other. So ask yourself, what failed?
Cytokinesis failed to divide the cytoplasm equally?
Right. If cytokinesis failed, what structure was damaged? the actin-myosin contractile ring, which relies heavily on myosin 2 interacting with the actin cortex.
Ah, I see.
You trace the clinical observation directly back to a molecular motor protein malfunction. That is incredibly satisfying. You aren't just shrugging and calling it a bad embryo. You are diagnosing a specific structural failure.
Exactly.
So let's apply that exact diagnostic mindset to the environment we force these gametes into. Let's talk about the hazards in the ART laboratory, because a petri dish is a highly controlled but ultimately very flawed simulation of the human reproductive tract.
It's an entirely imperfect simulation. And every single variable that deviates from physiological conditions, whether it's temperature, pH, osmolarity, has a specific, highly vulnerable molecular target inside the cell.
Let's start with the most obvious one, temperature. We are obsessed with 37 degrees Celsius, heating stages, tube warmers, incubators. But what is the actual intracellular target if the temperature drops? Say the dish is on an unheated part of the bench and the media drops below 35 degrees. What is physically breaking?
The primary casualty of thermal stress is the M2 spindle. The meiotic spindle is constructed of microtubules, which are polymers of alpha and beta-tubulin dimers. These microtubules are governed by a principle called dynamic instability. They are constantly growing and shrinking.
Like a scaffold that is constantly being built and torn down at the same time.
That's a perfect way to picture it. And that dynamic instability relies on a delicate thermodynamic equilibrium, specifically maintained by a GTP cap at the growing end of the microtubule.
And when the temperature drops?
When it drops below 35 degrees, that thermodynamic energy is just lost. The GTP cap hydrolyzes too quickly, and the entire microtubule undergoes catastrophic detelomerization. The spindle simply dissolves.
Wow. So the structural cables holding the chromosomes perfectly in place at the metaphase plate just vanish.
Yes. And the immediate consequence is chromosome scattering. The chromosomes drift away from the equator. Now, if you realize your mistake and quickly put that oocyte back into the 37-degree incubator, the spindle will eventually reform. Tubulin will repolymerize.
But I'm guessing it doesn't just snap back perfectly into place like nothing happened.
It rarely does. When it reforms, it frequently fails to capture all the scattered chromosomes correctly. You get kinetochores that aren't attached, or merotellic attachments where a single chromosome is literally being pulled in two directions.
Which means?
The clinical outcome of that brief temperature drop is non-disjunction during the anaphase transition. You have directly caused aneuploidy.
So a cold bench doesn't just make the oocyte sluggish, it actively scrambles its genetic sorting machinery. That completely changes how I view leaving a dish out for an extra 30 seconds.
It should.
What about pH? We keep our media tightly buffered, usually targeting an internal pH of about 7.2 to 7.4. What happens molecularly if the pH drifts outside that window?
Well, cellular proteins are exquisitely sensitive to hydrogen ion concentration. Their three-dimensional folding relies on specific electrical charges across their amino acids. If the pH drops too low, which is intracellular acidosis, or drifts too high, you alter the protonation states of these proteins.
Meaning their physical shape changes because their electrical charges are shifting.
Exactly. And when an enzyme's shape changes, its kinetic activity plummets. The primary targets here are cyclin-dependent kinases, or CDKs, which are the master regulators of the cell cycle.
So if pH drifts, CDK activity stalls, and the embryo just arrests.
Correct. Furthermore, pH heavily impacts calcium handling. The smooth endoplasmic reticulum relies on ion pumps to sequester calcium. If the pH alters the efficiency of those pumps, you get aberrant intracellular calcium levels.
Which is dangerous.
Very. It can trigger premature activation or even apoptosis.
Let's talk about osmolarity. Evaporation is a constant threat if oil overlays aren't perfect. If the media concentrates and osmolarity spikes to, say, 300 or 310 milliosmoles, water's going to leave the cell. The cell shrinks.
Right, osmotic dehydration. But the damage isn't just about losing water. It's about what that physical shrinkage does to the internal scaffolding. When the cell volume rapidly decreases, the plasma membrane collapses inward, compressing the underlying actin cortex.
It's like letting the air out of the dome tent. The poles just get crushed together.
That's a great analogy. You get severe actin disruption. And because the actin network is required for organelle transport, an immediate symptom of hyperosmolar stress is the arrest of cytoplasmic streaming.
Wait, what does that mean practically?
The internal transport network, like the delivery trucks moving mRNAs and proteins around the cell, just grinds to a halt. The cytoplasm basically freezes in place.
Okay, let's look at ambient atmospheric threats. Oxygen and light. We breathe air with roughly 21% oxygen, but the human fallopian tube and uterus are practically hypoxic by comparison, sitting around 2 to 8%.
Culturing embryos in 21% atmospheric oxygen is one of the most historically damaging things our field used to do. Atmospheric oxygen generates an overwhelming amount of reactive oxygen species, or ROS.
And where does that hit the hardest?
Inside the cell, the primary targets for ROS are the mitochondria and the plasma membrane.
Let's dig into the membrane aspect because this is fascinating chemistry. The plasma membrane is loaded with polyunsaturated fatty acids, right? PFAs. Why are they so vulnerable to oxygen?
Well, polyunsaturated fatty acids have multiple double bonds in their carbon chains. These double bonds are structurally necessary to keep the membrane fluid and flexible at body temperature.
Okay.
But chemically, the carbon-hydrogen bonds adjacent to these double bonds are highly reactive. When a hydroxyl radical, which is a highly aggressive form of ROS, bumps into a PUFA, it steals a hydrogen atom.
It rips it right off the chain.
Yes, initiating a vicious chain reaction called lipid peroxidation. The damaged lipid radical attacks the next lipid, and so on. But the real structural damage comes from the secondary byproducts of this reaction.
What are they?
The degradation of these lipids produces highly toxic aldehydes, primarily malondialdehyde, or MDA, and 4-hydroxynonal, or 4-HNE.
MDA and 4-HNE. What exactly do these toxic byproducts do inside the oocyte?
They act as cross-linking agents. They form covalent bonds between different lipids and between lipids and membrane proteins. They essentially bolt the fluid mosaic together.
Oh, wow. I always think of oxidative stress like rust. It's the slow chemical burn that stiffens up moving parts. If 4-HNE is bolting the lipids together, it's like pouring epoxy into the membrane. It loses all of its fluidity.
That is precisely what happens. And if the plasma membrane is stiff like old wax, what fails? First, gamete fusion fails.
Right.
For a sperm to fuse with an oocyte, the ulemma must be incredibly fluid to allow the proteins to undergo conformational changes and form a fusion pore. If the membrane is cross-linked and rigid, the sperm just bounces off.
And even if fertilization does somehow happen, what does a stiff membrane mean for cell division?
It means cytokinesis fails. When the contractile ring tries to pinch the cell in half, the membrane has to fold and stretch dynamically. A cross-linked, rigid membrane will resist that tension, leading to chaotic, fragmented cleavage or total cleavage arrest.
Man, so that's the membrane rust. What does ROS do to the mitochondria?
It attacks mitochondrial DNA, or mtDNA. Keep in mind, mitochondrial DNA is circular and lacks the protective histone proteins that shield nuclear DNA. Plus, it is located physically right next to the electron transport chain, which is the primary source of the cell's endogenous ROS.
It's sitting right next to the fire.
Constantly. It causes strand breaks and base modifications. This damages the inner mitochondrial membrane where the oxidative phosphorylation machinery lives. The clinical result? The ATP output of the mitochondria just plummets.
And this is uniquely devastating for an oocyte, right? Because they don't make new mitochondria.
Exactly. An oocyte inherits a massive pool of mitochondria, usually between 100,000 and 1 million. But mitochondrial replication is strictly shut down during early embryogenesis.
Wait, really? It's totally shut down?
Totally. The embryo must coast entirely on that maternal supply until it reaches the blastocyst stage. If ROS damages a significant portion of that pool, the embryo simply runs out of energy to drive the sodium pumps for blastical expansion, and it arrests.
What about the sperm? Does ambient ROS affect the sperm lying in the dish?
Severely. Because a mature spermatozoon has almost no cytoplasm, it has virtually no endogenous antioxidants to protect itself. ROS attacks the nuclear DNA in the sperm head, leading to the oxidation of guanine bases to form 8-oxoguanine.
8-oxoguanine. And that causes strand breaks.
It causes transversions during DNA replication, but it also physically destabilizes the chromatin structure, leading to single and double strand breaks.
Which we test for, right?
Yes, clinically we measure this as an elevated DNA fragmentation index. When an oocyte is fertilized by a sperm with high DNA fragmentation, the oocyte's DNA repair machinery has to work overtime to fix the breaks before the first cell division. If the damage exceeds the oocyte's repair capacity, development arrests.
This is exactly why the lab protocols are so incredibly rigid. We culture in 5% oxygen to starve the ROS generation process. We supplement media with antioxidants to scavenge whatever radicals do form. But what about the oil overlays? Is mineral oil just a physical lid to stop evaporation?
It is a physical lid, but it also acts as a chemical sink. Mineral oil can actually absorb certain volatile organic compounds and even some reactor species from the culture media.
That sounds good, though.
It is, but it's a double-edged sword. If the oil itself is exposed to UV light or heat during storage, It can undergo peroxidation before you even use it.
So you could be overlaying your embryos with a toxic peroxidized blanket.
Precisely. Which is why oil must be rigorously dark-stored and rigorously tested.
We didn't mention the other ambient room hazard visible light. If I'm working at the hood, just the normal microscopes and room lights, how much damage are they doing?
Visible light causes photooxidation. It excites photosensitive molecules in the cytoplasm and in the culture, media like riboflavin and certain amino acids.
Okay.
When these molecules absorb light energy, they enter an excited triplet state and transfer that energy to oxygen, creating singlet oxygen, which is a highly reactive ROS.
So light literally creates ROS out of thin air.
Out of the dissolved oxygen in your media, yes. This is why we insist on dimming the room lights, turning off the microscope light whenever we aren't actively looking, and using red or green filters that eliminate the more energetic, damaging blue wavelengths.
Let's shift from the ambient environment to the actual procedures. Let's trace an oocyte's journey through an ART cycle, starting with ovarian stimulation and the retrieval itself. Because the biology of making the egg is completely disrupted by our pharmacology.
During a natural cycle, the growth of the follicle and the maturation of the oocyte are perfectly synchronized. But in controlled ovarian stimulation, we are using exogenous gonadotropins to rescue a whole cohort of follicles and force them to grow simultaneously.
Right.
This creates a profound biological disconnect between nuclear maturity and cytoplasmic maturity.
This is a huge concept. I mean, an embryologist looks down the microscope, sees a first polar body, and says, great, it's an M2, it's mature. But you're saying the nucleus might be ready, but the cytoplasm is not.
Correct. The extrusion of the first polar body merely confirms that homologous chromosomes have separated. That's nuclear maturity. But cytoplasmic maturity refers to a massive, invisible reorganization.
Like what?
Well, the mitochondria must migrate from the periphery to the perinuclear space. The cortical granules must travel outward to line up perfectly under the ulema. The cytoplasm must accumulate massive stockpiles of maternal mRNA to sustain early development.
And if that doesn't happen?
If the stimulation protocol was too aggressive, the nucleus reaches M2, but the cytoplasm is left fundamentally unprepared.
So it looks perfect, but it's empty inside. And then comes the actual aspiration, the retrieval. You are jabbing a needle into the ovary and sucking the fluid out. We already discussed the thermal threat to the M2 spindle, but what about the physical physics of aspiration?
The physical shear stress is immense. If the aspiration pressure is too high, say, exceeding 150 millimeters of mercury, the velocity of the fluid moving through the needle creates extreme turbulence. This shear force can physically dismantle the delicate meiotic spindle just through mechanical vibration.
It literally shakes the spindle apart.
It does, and worse, that mechanical shock can displace the cortical granules away from the plasma membrane. If the granules aren't docked at the membrane, the oocyte cannot mount a proper cortical reaction to block polyspermy during IVF.
Okay, so we get the oocyte to the lab. If we are doing ICSI, we have to strip the cumulus cells off, denudation. We expose it to an enzyme, hyaluronidase, and we mechanically pipette it. What are the intracellular hazards here?
Chemically, hyaluronidase is used to digest the hyaluronic acid matrix holding the cumulus cells together. But if the concentration is too high or the exposure time is too long, the enzyme begins to act on the glycoproteins of the ulema itself.
So it starts digesting the egg membrane.
Exactly. It becomes fundamentally cytotoxic,
Right.
altering the membrane's permeability and making it highly susceptible to degeneration.
And the mechanical pipetting. Stripping the cells off by pulling them through a glass capillary.
The ulema is structurally anchored to the underlying actin cortex by various linker proteins, including myosin 1. When you force an oocyte through a stripper tip that is too narrow, the friction pulls on the membrane.
Oh, that sounds rough.
It is. This shear stress can literally tear the ulema away from the actin cortex. You disrupt the localized consanguinity of the cell, which can lead to spontaneous activation or immediate lysis.
Which brings us to ICSI itself, intracytoplasmic sperm injection. We are taking a sharpened glass pipette and driving it physically through the zona pellucida, through the plasma membrane, and deep into the cytoplasm. What is the cell experiencing during that intrusion?
It is experiencing a localized structural earthquake. The needle transiently but profoundly ruptures the cortical actin network.
Right.
As it pushes inward, it forms a deep invagination of the membrane before it finally pierces. This physically displaces organelles, mitochondria, and endoplasmic reticulum are just shoved aside.
And when you break the membrane, isn't there a massive influx of whatever is outside the cell?
Yes, there is a massive influx of extracellular calcium from the culture media through the breach. The oocyte must rapidly deploy repair mechanisms, often involving rapid exocytosis of local vesicles, to patch the hole before the calcium wave triggers premature, chaotic activation of the entire cell cycle.
Wow.
Furthermore, if you aspirate too aggressively to break the membrane, you draw significant amounts of cytoplasm into the needle. This violent fluid movement can rip the M2 spindle right out of position.
Let's talk about the specific topography of that spindle, because this leads directly to one of the most famous clinical rules in embryology, the 3 o'clock rule for ICSI. Why do we care exactly where the needle enters?
Because we must avoid the M2 spindle at all costs.
Okay.
The spindle is invisible under standard bright field microscopy, but we know its geographical location based on the first polar body. The polar body is tethered to the ulema, and the active meiotic spindles almost always located directly beneath the cortex, intimately associated with that polar body.
So if we imagine the oocyte as a clock face, and we orient the polar body at 12 o'clock, the spindle is sitting right there under the surface at 12 o'clock.
Exactly. If you were to inject at 12 o'clock, or even 1 or 11, the glass needle would spear directly through the microtubule array, completely obliterating the chromosomal alignment.
So by injecting exactly at the 3 o'clock position, or 9 o'clock,
You are entering 90 degrees away from the spindle. You maximize the physical distance from the delicate chromosomal machinery, ensuring that the shear forces of the injection fluid don't perturb the metaphase plate.
Let's stay on ICSI for a moment and talk about the snap-through. When I watch experienced embryologists do ICSI, they talk about the feel of the membrane. What are they actually sensing?
They are evaluating the viscoelastic properties of the ulema and the actin cortex. When you push the needle against a healthy oocyte, it doesn't just puncture, it indents deeply.
Right, like pushing into a water balloon.
Yes, the membrane stretches, forming a funnel around the needle. This shows elasticity. Then, with a little negative pressure or a slight tap, it suddenly breaks with a distinct snap-through.
And if it doesn't snap, if it just punctures immediately like a thin balloon,
That indicates a catastrophic loss of membrane elasticity. The membrane has become rigid, usually due to the exact oxidative stress and lipid peroxidation we discussed earlier.
the 4-HNE rust.
Exactly. Or perhaps overexposure to hyaluronidase. A rigid membrane is brittle. Conversely, if it stretches indefinitely and never snaps, it indicates a breakdown of the underlying actin scaffolding. Both extremes are strong indicators of prior cellular trauma and correlate with much lower survival rates.
Moving to the last major procedural hazard, cryopreservation, freezing and thawing. We touched on osmotic shock, but I want to understand the exact mechanics of vitrification. How do we freeze a cell without destroying it with ice?
Water expands when it crystallizes into ice. If ice crystals form inside a cell, they physically spear the organelles and shred the plasma membrane. It's lethal. Vitrification avoids this by turning the intracellular fluid not into ice, but into a glass-like amorphous solid.
How do you turn water into glass?
By replacing a massive percentage of the intracellular water with high concentrations of cryoprotectants, like ethylene glycol and DMSO, and then plunging it into liquid nitrogen so fast that crystals literally don't have time to form.
How fast are we talking?
The cooling rate is over 20,000 degrees Celsius per minute.
That is insane. But getting all that water out and all those chemicals in must put immense strain on the cell.
It is the ultimate osmotic stress test. When you move an embryo into the equilibration solution, the external osmolarity skyrockets. Water rushes out of the cell instantly.
Right.
The cell violently shrinks, plasmolizing away from the zona pellucida. The internal organelles are basically crushed together.
And what determines if it survives that crush?
The efficiency of its water channels, specifically aquaporins. Aquaporin 3 and aquaporin 7 are vital here. They facilitate the rapid efflux of water and the influx of the cryoprotectant.
So if those aren't working,
If the aquaporins are malfunctioning, the fluid shifts take too long, the cell doesn't dehydrate adequately, and microscopic ice forms during the plunge. Furthermore, the cytoskeleton must be resilient enough to survive the compression and then physically re-expand during the warming process without the actin filaments permanently tangling or breaking.
it's miraculous that it works at all, honestly. So we've navigated the hazards, we've kept the temperature stable, minimized ROS, and injected the sperm perfectly at 3 o'clock. Now we have to evaluate what we've got. Let's move into morphology assessment based on the ESHRE and LFHA consensus criteria. This is where we translate cellular realities into clinical scores.
Correct. And to do this systematically, we divide the dysmorphisms of an oocyte into two broad categories: Intracytoplasmic meaning inside the main body of the cell, and extracytoplasmic.
Let's start intracytoplasmic. If I'm looking at an oocyte and the cytoplasm isn't smooth, if it has central or diffuse granularity, what is functionally happening? What are those granules?
You are seeing the physical clustering of organelles, primarily mitochondria, and the endoplasmic reticulum. In a highly competent oocyte, mitochondria are smoothly and evenly dispersed throughout the cytoplasm to provide a uniform supply of ATP, especially to the energy-hungry molecular motors along the cortex.
So granularity means the power plants have clumped together.
Yes, and when they cluster, they leave massive dead zones in the cytoplasm that are entirely starved of ATP. This clustering is generally a downstream effect of cytoskeletal breakdown. The microtubule tracts that hold them in place have degraded. It's a sign of cellular aging or severe in vitro stress.
What about refractile bodies? These are the bright, shiny inclusions you sometimes see catching the light.
Refractile bodies are essentially biological trash. They are dense aggregates of oxidized lipids and denatured proteins. Biochemically, they are highly analogous to lipofuscin, the wear and tear pigment that accumulates in aging neurons and heart muscle.
Wait, so these shiny spots are literally the cross-linked MDA and 4-HNE complexes we talked about during lipid peroxidation.
Exactly. When the cell's autophagic machinery tried to clean up oxidatively damaged membranes, but fails to completely digest them, it walls them off into these dense, insoluble granules. A refractile body is a microscopic tombstone of a severe ROS attack.
That makes it so much more vivid than just calling it a shiny spot. What about vacuoles? You know, the empty-looking bubbles inside the cell.
Vacuoles usually represent massive dilatation of the smooth endoplasmic reticulum, or the runaway accumulation of autophagosomes. Small vacuoles are common and often benign, but large vacuoles, specifically those greater than 14 micrometers in diameter, are a severe warning sign.
Why does size matter so much there?
Because a massive vacuole displaces the entire internal architecture. It pushes the spindle out of alignment. More importantly, if an embryologist accidentally ruptures a 14-micrometer vacuole with the ICSI needle, the sudden release of its contents, which are often acidic or enzymatically active, into the general cytosol is immediately lethal to the oocyte.
Okay, here is one of the most critical exam notes and clinical pivots of the last decade. Smooth ER aggregates, or SARA. For a long time, if an embryologist saw SARA, that oocyte went straight in the bin.
It was considered the absolute kiss of death. Early studies linked SARA to catastrophic birth defects, specifically Beckwith-Wiedemann syndrome. Laboratories established strict auto-discard policies. But as larger multi-center data sets emerged, the field realized those early correlations were statistically flawed.
So what is the current ESHRE consensus?
The guidance is emphatically clear. Do not auto-discard oocytes with SARA.
I need to understand why. What actually is in SARA and why is it safe?
SARA stands for a smooth endoplasmic reticulum aggregate. It is a large, flat, disc-like clustering of smooth ER tubular networks. Functionally, the smooth ER is the cell's calcium vault. So in SARA represents a spatial misallocation of calcium stores.
Which sounds bad because calcium is the master trigger for fertilization.
It is a dysmorphism. It suggests a mild disruption in calcium homeostasis. However, the data proves that in the vast majority of cases, the oocyte's overall calcium release machinery is robust enough to compensate for this localized clustering. The calcium wave still propagates. Fertilization still occurs.
Wow.
And these oocytes regularly result in healthy, euploid live births. Discarding them is just a tragic waste of reproductive potential.
What about large lipid droplets? I know we see these more frequently in oocytes from patients with high BMI or metabolic syndrome.
Yes, large lipid droplets indicate an excess accumulation of neutral lipids, specifically triglycerides. While a certain amount of lipid is normal and used for energy, massive droplets indicate lipotoxicity.
What does that do?
The excess lipids induce endoplasmic reticulum stress and trigger apoptotic pathways, severely impairing the oocyte's ability to mature or survive fertilization.
Okay, let's move outside the plasma membrane. Extracytoplasmic dysmorphisms. The most obvious structure is the zona pellucida. It's the protective shell. What does it mean if the zona is unusually thick or dark?
The zona pellucida is a matrix of specific glycoproteins, ZP1, ZP2, ZP3, and ZP4. These are actively synthesized and secreted by the growing oocyte during folliculogenesis. A thickened or excessively dark zona indicates a profound disruption in that secretory pathway.
Meaning?
It means the Golgi apparatus was overactive or the timing of transcription was flawed. Clinically, a very thick zona can impair the physical hatching of the blastocyst later on, though we can bypass that with laser-assisted hatching.
What about the paravitaline space, the PVS? That's the gap between the cell membrane and the zona. If that space is massive or filled with granular debris,
A massively enlarged PVS often indicates that the oocyte has shrunk, which points to osmotic stress or chronic metabolic compromise. If it's full of granular debris, those are often prematurely exocytosed cortical granules.
meaning the oocyte essentially panicked and fired its anti-polyspermy weapons before the sperm even arrived.
Precisely.
And a fragmented first polar body. Yeah, that's a common one. Does a shattered polar body mean the egg is genetically doomed?
Not at all. The polar body is a terminal structure. It is destined to degrade anyway. Fragmentation simply means that the extrusion process during meiosis I, which requires intense contractile ring activity, was physically stressful, causing the polar body to bleb apart.
So it's not a deal breaker.
It suggests the cytoskeleton was under strain, but alone it does not disqualify the oocyte from making a perfect embryo.
So ICSI is done. We've assessed the egg, we've injected the sperm, and we put the dish in the incubator. Let's trace the developmental timeline. Day 1, which we check at 16 to 18 hours post-ICSI. The absolute ideal visual is 2 PN, 2 pronuclei, and 2 polobodies.
Right. That confirms normal monospermic fertilization. The sperm delivered its payload, the oocyte resumed meiosis, extruded the second polar body to ditch the extra chromosomes, and now the male and female genomes are unwinding into these large visible pronuclei.
But ESHRE expects us to look far deeper than just counting 2 circles. We need to look inside the pronuclei at the nucleolar precursor bodies, the NPBs. What exactly is an NPB?
The nucleolus is the region of the nucleus where ribosomes are assembled. In a newly fertilized zygote, transcription hasn't started yet, so these aren't true functional nucleoli. They are dense clusters of proteins and RNA waiting to be activated,
Okay.
hence nucleolar precursor bodies.
What do they look like?
They look like dark, distinct pinpricks inside the pronucleus.
And what are we looking for them to do?
We want to see perfect polarization. We want an equal number of NPBs in each pronucleus, usually between 3 and 7, and we want them lined up perfectly parallel to each other, exactly along the junction with the 2 pronuclei you're touching.
Okay.
This is classified as a Z1 pattern in the Z-score system, or a high Scott score.
I understand it looks neat and tidy, but mechanically, why does it matter where these dots line up?
Because the NPBs are physically tethered to the chromatin, the DNA itself. Their movement is driven by the vast microtubule network orchestrated by the sperm's centrosome.
Ah.
If the NPBs are polarizing symmetrically to the junction, it is visual confirmation that the molecular motors and the cytoskeletal scaffolding are functioning flawlessly. They are actively pulling the maternal and paternal genomes into perfect spatial alignment so that when the pronuclear membranes break down, the chromosomes will intermingle perfectly on the first mitotic spindle.
So NPB polarization is essentially watching the gears of syngamy locking into place. If the NPBs are scattered randomly, it means the cytoskeletal towing cables are broken or chaotic.
Exactly. The spatial organization predicts the accuracy of the upcoming division.
Which brings us to day 2, 43 to 45 hours post-ICSI. We open the incubator, or check the time-lapse, and we expect to see a 4-cell embryo. The classic criteria are blastomere symmetry and less than 10% fragmentation. What does perfect symmetry tell us about the intracellular mechanics?
Blastomere symmetry confirms the spatial precision of the mitotic spindle. In a normal division, the centrosomes migrate to exactly opposite poles of the cell, building a perfectly centered bipolar spindle. The contractile ring forms precisely at the equator.
So it cuts it right down the middle.
Yes. When it pinches, it divides the cytoplasm exactly 50-50. If the blastomeres are dramatically unequal, it means the spindle was anchored off-center, pointing to a failure in the actin microtubule crosstalk.
And fragmentation, we mentioned this earlier, but I want to clarify the mechanism. Fragments look like tiny little cells without nuclei. How do they form?
Fragmentation is essentially unregulated membrane blebbing. During cytokinesis, the cortex is under massive tension from myosin II pulling on actin filaments. If the actin network is locally damaged, perhaps by the ROS we discussed, the localized tension snaps. The membrane bulges out and pinches off entirely, taking a chunk of cytoplasm with it. It's an amputated piece of the cell.
That's a brutal way to picture, but very accurate. Let's talk about a major day 2 red flag, multinucleation. Seeing 2 or 3 nuclei trapped inside a single blastomere. How does a cell end up with extra nuclei?
Multinucleation represents a profound failure to couple karyokinesis with cytokinesis.
Explain the difference between those two terms for a second.
Karyokinesis is the division of the nucleus. The chromosomes replicate, the spindle pulls them apart, and two new nuclear envelopes form around them. Cytokinesis is the division of the cytoplasm, the actual physical pinching of the whole cell into two.
So in multinucleation,
The cell successfully copied and separated as DNA karyokinesis worked, but the contractile ring failed. Cytokinesis never happens. The cell didn't split.
oh,
So now you have a single giant blastomere containing double the amount of genetic material housed in multiple nuclei. The gene dosage is completely chaotic.
moving to day 3, 67 to 69 hours. We expect to see 7 to 9 cells. But if we are using time-lapse morphokinetics, we get to see how it reached that cell number. I've always struggled to visualize direct unequal cleavage, or DC. How does a single cell mathematically skip the 2-cell stage and divide directly into 3 cells all at once?
It defies normal geometric logic because it is a catastrophic structural failure. In a normal division, a cell has 2 centrosomes that organize a bipolar spindle. The chromosomes align in a single line and are pulled to 2 poles.
Right, one splits into 2.
In direct unequal cleavage, the embryo has inherited or generated supernumerary centrosomes, meaning 3 or more organizing centers. This causes the formation of a multipolar spindle.
Wow.
The chromosomes don't form a single line. They are arranged in a chaotic Y shape or a triangle. When anaphase triggers, the chromosomes are physically ripped in 3 different directions simultaneously. The contractile ring tries to follow this chaos and pinches the cell into 3 highly unequal, genetically chaotic blastomeres.
Are the chromosomes literally physically tearing?
They can be. Meritelic attachments, where a single kinetochore is pulled by microtubules from 2 different poles, often result in chromosome breakage or lagging chromosomes that get lost entirely. An embryo with DC is almost guaranteed to have massive, complex aneuploidy.
What causes a day 3 arrest? Say the embryo looks beautiful on day 2, perfect symmetry, no fragmentation. But by day 3, it just stops. It never moves past 6 or 8 cells.
That specific timeline points directly to a failure in zygotic genome activation, or ZGA. Up until the 4 to 8 cell stage in humans, the embryo is completely transcriptionally silent.
Meaning it's not using its own DNA yet.
Right. It is coasting entirely on the maternal mRNA that the oocyte stockpiled during folliculogenesis.
It's flying on battery power.
Exactly. And right around day 3, 2 things must happen simultaneously. First, the maternal mRNA must be actively degraded. This is done via deadenylation, where deadenylase enzymes systematically chop off the poly A tails of the maternal transcripts, silencing them.
Okay, so step 1 is shutting off the old instructions.
Yes. Second, the newly combined embryonic genome must wake up and start transcribing its own DNA.
The new engine has to catch.
Yes. If the maternal transcripts are degraded, but the embryonic genome fails to activate, perhaps due to heavy epigenetic methylation that wasn't cleared, the embryo suddenly has no mRNA to make proteins. The biochemical machinery halts. The embryo arrests precisely on day 3.
So if the engine catches, the embryo survives day 3 and makes a massive leap to day 5 or 6. It undergoes compaction, and then it must become a blastocyst. This is the ultimate checkpoint evaluated using the Gardner grading system.
And this is where ESHRE integration is paramount. The Gardner system assigns a number for expansion and 2 letters for the cellular compartments. You must understand that these aren't just aesthetic grades. They reflect highly complex, distinct intracellular mechanics.
Let's deconstruct it. The first parameter is the number, 1 through 6, indicating the degree of blastical expansion. What is literally inflating that cavity? What is the pump?
The pump is the sodium-potassium ATPase, localized specifically to the basolateral membranes of the trophectoderm cells. When compaction occurs on day 4, the outer cells polarize. These ATPase pumps actively push sodium ions into the central intracellular space.
And where sodium goes, water follows.
Exactly. Osmosis draws water from the surrounding media into the embryo to dilute that sodium. But water can't cross the lipid bilayer fast enough on its own, so this expansion is highly dependent on aquaporin channels opening up to flood the cavity.
So a grade 1 is an early blastocyst where the ATPase pumps are just turning on and the first droplets of water are coalescing.
Right. And by grade 3, you have a full blastocyst. The cavity is fully inflated, pressing against the zona pellucida. Now, for that cavity to hold hydrostatic pressure without leaking, the trophectoderm cells on the outside must be sealed perfectly tight.
Like caulking a bathtub.
Yes, exactly. That cellular caulking consists of tight junctions, formed primarily by transmembrane proteins called claudins and occludins, and they're anchored internally by a very strong circumferential actin ring.
And if they fail?
If the tight junctions fail, the water leaks out as fast as the pumps push it in, and the blastocyst just collapses.
And by grade 5, the pressure is so great and the trophectoderm has secreted specific lytic enzymes that the embryo bursts out of the zona pellucida. It's hatching.
Correct.
Okay, the next part of the grade is a letter, A, B, or C, evaluating the inner cell mass, the ICM. This is the cluster of cells that will actually become the fetus.
A grade A ICM is a tightly packed, prominent mass with many cells. Intracellularly, this visual reflects high cortical tension, which physically maintains the cells in a round, compact state.
And molecularly?
Molecularly, it correlates with the robust expression of core pluripotency transcription factors, specifically OCT4 and NANOG. These genes keep the ICM cells undifferentiated and capable of forming every tissue in the human body.
What does a grade C ICM look like, and what does it mean?
A grade C ICM has very few cells, and they look loose and disorganized. This reflects a failure in epigenetic lineage allocation. During compaction, the inner cells failed to upregulate OCT4, or perhaps they're undergoing apoptosis due to accumulated DNA damage. A grade C ICM carries a strong adverse prognosis for fetal development. Even if it implants, it frequently results in a blighted ovum.
Finally, the third part of the grade, another letter for the trophectoderm, the TE. This is the outer sphere of cells.
The TE is absolutely vital because it becomes the chorion and ultimately the placenta. A grade A TE shows a cohesive, continuous epithelium with many cells. It reflects intact tight junctions, healthy ATPase activity, and robust expression of the CDX2 transcription factor, which is the master regulator of trophectoderm fate.
I feel like embryologists often hyper-focus on the ICM because it becomes the baby, but the TE is the life support system. A bad TE grade is a critical failure point.
Absolutely. The TE is what physically invades the endometrium and establishes maternal blood flow. If the TE fails to implant or fails to form a functional placenta, the pregnancy fails entirely, regardless of how biologically perfect the ICM was.
Okay, we have assessed every stage. We have all this rich morphological data. Now comes the hardest part of the job, making the call. Embryo selection. And ESHRE enforces a massive mindset shift here.
They do. ESHRE mandates that embryologists transition away from binary categorical labeling like calling an embryo good or bad, normal or abnormal, and move toward probabilistic ranking.
Why is that distinction so important?
Because biology is a spectrum, not a binary. If you see a single refractile body in an oocyte or a 5% fragmentation on day 2, it does not mean the embryo is biologically broken or genetically doomed. It simply means that statistically, its probability of live birth is slightly lower than a visually perfect sibling cohort. You rank them by cumulative developmental potential.
So what is the hierarchy of that ranking? If I have a cohort of 5 blastocysts, what data point holds the supreme weight?
Number 1 is universal euploidy. If you have pre-implantation genetic testing for aneuploidy PDTA results, a euploid embryo overrides almost all morphological concerns.
Right, because if the genetics are fundamentally unbalanced, the prettiest TE in the world won't prevent a miscarriage. What's number 2?
Number 2 is the blastocyst grade, the Gardner expansion, ICM, and TE scores. Number 3 is morphokinetics data from the time-lapse, looking for anomalies like direct unequal cleavage or reverse cleavage.
Got it.
Number 4 is day 1 pronuclear morphology. Did it have a perfect Z1 NPB pattern? And the lowest weight, number 5, is the initial oocyte morphology.
So an oocyte with a thick zona, a slightly granular cytoplasm, and maybe even a small vacuole could still rank as your number 1 transfer choice if it forms a euploid 4AA blastocyst.
Precisely. The outcome overrides the origin.
Let's do a mock clinical case review. I'll give you a specific bench observation, and I want you to give me the ESHRE aligned clinical action and the specific molecular mechanism behind it. You check fertilization on day 1, 16 hours post-ICSI. You look down the microscope and see 3 p.m., 3 pronuclei.
Action. Discard immediately. Mechanism. The zygote is triploid, meaning it has 69 chromosomes instead of 46. Since it was ICSI, we know multiple sperm didn't enter. Therefore, the mechanism is the oocyte failed to extrude the second polar body upon activation.
So it kept an extra set of chromosomes.
Exactly. The maternal genome copied itself, but the contractile ring failed to throw away the extra set. It retained 2 maternal pronuclei plus the paternal one. It is genetically incompatible with life.
Next observation. You check at 16 hours and you see 0 p.m. No pronuclei visible, just a clear cytoplasm.
Action. Do not discard. Observe and culture forward.
Hey, really? Why?
Mechanism. There are 2 distinct possibilities. The most common is failed fertilization. The sperm failed to trigger the calcium wave. But the second possibility is early syngamy. You simply checked too late. The pronuclei formed perfectly at 10 hours, and by 16 hours, the nuclear envelopes had already broken down to prepare for the first cleavage. If you discard it, you might be throwing away a perfectly healthy, fast-developing embryo.
Okay, observation. Day 1, you see one PN, but you specifically notice an absent second polar body.
Action. Suspect profound activation failure. Keep in culture, but lower its rank severely. Mechanism. The 1PN is likely just the female chromatin unwinding. The absent second polar body proves the oocyte never received the downstream calcium signal required to activate CAMK2.
Remind me what CAMK2 does again.
CAMK2 is the enzyme that destroys maturation-promoting factor to resume meiosis. Without it, the polar body stays trapped. For this patient's next cycle, you must strongly consider artificial oocyte activation using a calcium ionophore.
Let's pivot to something that requires a completely different but equally vital skill set, patient counseling. All of this brilliant science, the CAMK2 pathways, the lipid peroxidation, means absolutely nothing if you sit down with a patient and panic them with isolating jargon.
This is a major testing point for ESHRE. A clinical embryologist must translate complex molecular failures into language that is scientifically accurate, deeply empathetic, accessible, and non-alarmist.
Let's workshop some of the exact verbiage. How do you explain the impact of maternal age on oocyte quality without just bluntly saying your eggs are old and dying?
You never use the word old. You focus on the physical mechanics. Instead of talking about lipid peroxidation and the cross-linking of PUFAs, you talk about flexibility. You say, as eggs wait in the ovary over time, their outer layers naturally accumulate wear and tear. They become a little less flexible.
That's a great way to put it.
You add, this stiffness makes it harder for the egg to easily fuse with the sperm and to divide smoothly.
Less flexible due to wear and tear. That perfectly conveys the mechanism of MDA and 4-HNE membrane stiffening without a biochemistry lesson. What about mitochondrial depletion?
We call them energy batteries. You say every egg carries a stockpile of microscopic energy batteries that have to power the embryo for the first 5 days. Over time, some of these batteries lose their charge. When an egg has fewer fully charged batteries, it might simply run out of the energy required to complete the massive task of cell division.
And aneuploidy, that word itself sounds terrifying to a patient.
You frame it around the machinery. To create a healthy embryo, the egg has to sort thousands of DNA threads perfectly. To do this, it builds a microscopic sorting machine. As we age, the parts of that sorting machine become less sticky and a little less reliable, so chromosomes occasionally get sorted into the wrong pile.
I really like that.
This is profoundly comforting to patients. It shifts the blame away from their behavior or their body rejecting a pregnancy and frames it exactly as it is. A microscopic mechanical error.
Let's practice delivering nuanced news. Say you have a cycle where you utilize several oocytes that had sera. The patient read online that this is a lethal defect because of old blog posts. How do you walk them off that ledge?
You normalize the observation using data. You say, when we looked closely at your eggs, we noticed a slightly unusual texture in the cytoplasm tiny clusters, called smooth ER aggregates. A decade ago, scientists were overly cautious about this texture. But today, analyzing thousands of global cases, we know definitively that these eggs have the exact same statistical chance of creating a healthy, beautiful baby as any other egg. So we confidently fertilize them.
What about fragmentation on day 2?
Again, normalize but inform. We see a small amount of fragmentation in the embryos today. Fragmentation is simply the cells shedding little bits of debris as they divide under the hard work of growing. It's incredibly common, like sawdust when cutting wood. We just factor the amount of sawdust into our priority ranking when deciding which embryo looks strongest for transfer.
And the hardest conversation, total failed fertilization after ICSI.
You must link the failure to the invisible biology so it doesn't feel arbitrary or mystical. Fertilization isn't just about putting the sperm inside the egg. It requires a very specific molecular key from the sperm to unlock the egg's growth cycle.
The PLCζ.
Exactly. When it fails, even with ICSI, it almost always means that specific microscopic key was missing or misshapen. But the good news is, now that we know the lock is stubborn, for your next cycle, we can use specialized solutions in the lab to help turn that lock artificially.
You link the biology to what the lab controls. That is deeply empowering. Okay, we are in the home stretch. We need to solidify the ESHRE exam matrix. These are the high-yield specific facts, names, and numbers that separate a passing score from a failing one. Let's dig into the specific themes.
The ESHRE blueprint tests precise recall. It does not have to know the general idea. You must know the actors and the measurements.
Theme 1, membrane structure and gamete fusion.
You must know the origins of the fluid mosaic model. proposed by Singer and Nicholson in 1972. You must know that the bilayer is amphipathic hydrophilic heads, hydrophobic tails. Critically, you must know the specific, recently discovered gamete fusion proteins.
Which are?
The sperm expresses a protein called IZUMO1.
IZUMO1, named after a Japanese marriage shrine, right?
Exactly. And IZUMO1 binds precisely to a receptor on the ulemma, called JUNO. If either IZUMO1 or JUNO is knocked out or sterically hindered by rigid membranes, fusion is mathematically impossible.
Step 2. Cholesterol and fluidity.
Cholesterol acts as a bidirectional fluidity buffer. It prevents the membrane from melting at high temps and prevents it from freezing at low temps. It comprises about 20% of the membrane lipid by weight.
Does this link to capacitation?
Yes. You must link this to sperm capacitation. For a sperm to undergo the acrosome reaction, it must actively efflux or pump out cholesterol from its membrane to increase fluidity enough for the acrosome vesicle to fuse with the plasma membrane.
Theme 3. Mitochondria numbers.
We stated this earlier, but memorize the bounds. 100,000 to 1 millisecond per mature oocyte. You must know the exact length of the human mitochondrial genome, 16,569 base pairs. And you must know that inheritance is strictly maternal.
Why strictly maternal? Don't sperm have mitochondria in their midpiece to power the tail?
They do, but upon entering the oocyte, the sperm mitochondria are ubiquitinated, meaning they are tagged with a death signal and actively destroyed by the oocyte's proteasomes. This prevents heteroplasmy, which is the chaotic mixing of two different mitochondrial genomes.
Theme 4. The trigger for activation. What is the specific molecular key we mentioned to the patients?
The key is a sperm-specific enzyme called PLCζ, phospholipase zeta. When the sperm enters, it releases PLCζ into the cytoplasm. This enzyme cleaves a membrane lipid called PIP into two products, D-shit and the critical messenger IP3.
And what does IP3 do?
IP3 diffuses through the cytoplasm and binds directly to IP3 receptors located on the smooth endoplasmic reticulum. This binding opens the calcium valves, triggering the massive cyclical calcium oscillations that break the meiotic arrest via camete T2.
Theme 5. The cytoskeleton and the spindle checkpoint.
First, know that the human meiotic spindle is a centrosomal. It forms without centrioles, organized entirely by microtubule organizing centers and motor proteins. Second, you must know the specific proteins of the spindle assembly checkpoint, the SAC.
What are the names?
MAD1, MED2, and BUBR1. These proteins bind to kinetochores that are not attached to a microtubule. They broadcast a weight signal that physically halts anaphase. If all kinetochores attach perfectly, the SAC proteins leave, the weight signal stops, and the cell divides.
So how does age affect this?
The clinical link is that as a woman ages, the expression of MAD1 and MAD2 naturally declines.
Meaning the weight signal gets weaker, the cell divides even if chromosomes aren't attached properly, leading straight to aneuploidy.
Precisely. You also need to know about cohesin, the protein rings that hold sister chromatids together. Cohesin is loaded during fetal life and never replenished. Over decades, cohesin rings naturally degrade. When they fail, chromatids drift apart prematurely.
Theme 6. The cortical reaction.
Cortical granules are modified lysosomes generated by the Golgi apparatus.
Right.
We must transport them to the membrane. The specific motor protein for this localized delivery is myosin 5A. Myosin 5A walks along the actin filaments carrying the granules to the cortex.
And when the calcium wave hits, they exocytose. What is the active ingredient inside them?
The primary active enzyme is ovostasin. When released into the perivitaline space, ovostasin specifically targets and cleaves the ZP2 glycoprotein in the zona pellucida. This cleavage fundamentally hardens the matrix, rendering it impenetrable to any additional sperm. It's the ultimate block to polyspermy.
Theme 7. Cilia and flagella structure.
Know the microtubule arrangement,
Mm-hmm.
the classic 9+2 axoneme structure. 9 peripheral doublets and 2 central singlets. Know that the motor protein driving the bending motion of the sperm tail is dynein. If a patient has a genetic mutation in the dynein arms, they suffer from primary ciliary dyskinesia.
Which means all the cilia in their body fail. They have chronic respiratory infections, and the clinical presentation in the fertility clinic is 100% immotile sperm, a direct, unavoidable indication for ICSI.
Exactly.
And finally, Theme 8. Telomeres.
Telomeres are the protective caps at the ends of chromosomes. You must know the exact nucleotide repeat sequence, TTAGGG. You must know that they're shielded from being recognized as DNA damage by a protein complex called shelterin, and know that telomerase, the enzyme that extends them, is highly active in the early embryo to reset the biological clock for the next generation.
TTAGGG and shelterin. Okay.
Yeah.
We have covered the hazards, the grading, the counseling, and the exam matrix. Now for the grand synthesis. This is where we tie the absolute bow on this curriculum. Let's run through 3 final complex worked examples of mechanistic reasoning, from the bench observation all the way down to the molecule.
I'm ready.
Case 1. You have a 32-year-old patient, excellent ovarian reserve, 15 mature M2 oocytes retrieved. You perform standard ICSI, injecting morphologically normal sperm. The next morning, you have 0 fertilization across all 15 eggs. Total fertilization failure.
Mechanistically, assuming the ICSI technique was flawless, the sperm physically breached the membrane, but the downstream biochemical cascade never initiated. This points to a failure at the very top of the activation pathway. The sperm was highly likely deficient in PLCζ.
So the sperm lacked the enzymatic key.
Yes, without PLCζ, no PIP2 is cleaved, no IP3 is generated. The IP3 receptors on the smooth ER are never stimulated. The massive calcium vaults remain locked. Because intracellular calcium levels never spike, CAMK2 is never activated.
So the cycle doesn't resume.
Right. Maturation promoting factor remains high, locking the cell in metaphase 2. The second polar body is never extruded, and pronuclei never form. The oocyte is paralyzed in a state of suspended meiotic animation.
Case 2. A 38-year-old patient. 8 fertilized zygotes. On day 2, they're all beautiful, symmetrical 4-cell embryos with 0 fragmentation. But on day 3, every single one of them arrests at the 6 to 8 cell stage. None of them compact on day 4.
The timing is the diagnostic key. This is a systemic zygotic genome activation failure. Up until the 4-cell stage, the embryo successfully utilized the maternal mRNA stockpiles. The ribosomes were translating proteins perfectly, but at the 8-cell stage, the transition must occur.
Right.
In this case, either the deadenylase enzyme successfully destroyed the maternal mRNA, but the embryonic genome failed to initiate transcription, or the embryonic genome was fundamentally too aneuploid to create coherent survival proteins. Without new transcripts, the molecular motors, the myosins, the tubulins, the ATPases cannot be replenished. The cellular machinery simply runs out of fuel and halts.
Case 3. A patient with recurrent implantation failure. She makes blastocysts, but every time you biopsy the trophectoderm and send it for PGTA, the results come back as chaotic complex aneuploidy, monosomies and trisomies across multiple different chromosomes.
Chaotic aneuploidy across multiple chromosomes is a hallmark of structural segregation failure, not a single non-disjunction event. Mechanistically, this points to a severe breakdown of the spindle tensegrity. It strongly implicates a severe decline in cohesin proteins, meaning the sister chromatids drifted apart before anaphase even began. It also implicates a critical failure of the spindle assembly checkpoint proteins, MAD1, MED2, and BBR1. The kinetochores were unattached, but the SAC proteins failed to generate the weight signal. The cell divided blindly, ripping the unaligned chromosomes into random, chaotic groupings.
It is so profoundly satisfying to connect the microscopic dots like that, to not just look at a failed embryo and feel helpless, but to be able to point precisely to a cohesin depletion, or a PLCζ absence, or a failed deadenylation pathway.
That is exactly the difference between a laboratory technician and a master clinical embryologist.
And that brings us to the complete picture. Over the course of this incredible curriculum, we have journeyed from the absolute atomic basics of lipid compartmentalization through the thermodynamics of the cytoskeleton, all the way to the specialized clinical decisions made at the bench regarding blastocyst expansion and patient counseling.
We've covered how the molecular saturation of a polyunsaturated fatty acid determines cryosurvival, how the spatial organization of the actin network dictates trophectoderm lineage, how the epigenetics of the inner cell mass control pluripotency. It is all one continuous, unbroken story of life.
It has been a monumental amount of information. As we wrap up this deep dive, do you have a final forward-looking thought to leave our listeners with? Something to ponder as they head into their ESHRE exams and back into the dark of the IVF lab?
I do. We have spent hours discussing how to visually assess these structures using bright field microscopy. But the human eye has absolute limits. We are squinting at shadows, trying to guess if the mitochondria are clustered or if the spindle is perfectly aligned. My provocative thought for you is this. Within the next decade, artificial intelligence and non-invasive hyperspectral imaging will likely bypass the human eye entirely.
Wow, bypass it entirely.
Yes, algorithms won't just look at the shape of a blastomere. They will map the metabolic heat signature of the mitochondria in real time. They will visualize the invisible spindle without fluorescent tags. They will literally see the echoes of the calcium waves and the tension of the molecular motors.
That is incredible.
The question for the embryologist of the future won't be, how do I score this embryo, but rather, how do I clinically interpret the massive molecular data map the AI just generated?
The machine will map the terrain, but the embryologist still has to navigate the journey. I love that. We are moving from observing biology to truly reading it. I want to thank you for guiding us through this incredible deep dive and for dismantling these dense concepts with such precision.
It has been an absolute privilege.
And to you, the listener, preparing for your ESHRE certification. Thank you for your immense dedication to mastering clinical embryology. We know it is not an easy path. The hours are long, the pressure is immense, and the stakes are literally life itself. But the knowledge you have synthesized here doesn't just pass difficult exams, it fundamentally builds human families. We wish you the absolute best of luck on your ESHRE examination and in your daily practice in the ART lab. Keep asking why, keep looking deeper into the biology, and we'll catch you on the next deep dive.
Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.